Welcome all to the penultimate DNA dialogue hosted by Australian Genomics. I'm Tiffany Bortwood. I'm the Managing Director of Australian Genomics. As we come together today in the spirit of reconciliation, I acknowledge the traditional custodians of country throughout Australia and the continuing connections to land, sea and community. I'm joining you from the lands of the Wurundjeri people of the East Kulin Nation. I pay my respects to their elders past and present and extend those respects to all Aboriginal and Torres Strait Islander peoples joining us today. So as you listen to the talk that we have today, please ask any questions you like during the talk using the Q&A function, and we'll do our best to get to as many of those as we can during the question time. If you are active on social media, please use the hashtag DNA Dialogue and tag AusGenomics as you're writing about this presentation today. So it's my very great pleasure to invite and introduce my friend Eva Maria Cutiongco-de La Paz to the presentation. Eva, you can turn on your camera now. There she is. So I'll give you a quick introduction. So Eva is a research professor at the Institute of Human Genetics and clinical professor of pediatrics and genetics at the University of the Philippines. She currently serves as the Executive Director of the National Institutes of Health. She finished her Doctor of Medicine from the University of the Philippines College of Medicine and her pediatric residency at the Philippine General Hospital. She had a research fellowship in molecular genetics at the International Center of Medical Research at Kobe University, Graduate School of Medicine in Japan, and took her clinical genetics training at Sick Kids in Toronto. Her research interests include genetics of monogenic and complex genetic conditions in the Filipino population. And today, Eva will be sharing her presentation entitled Investigating the Genetic Epidemiology of X-linked Dystonia Parkinsonism. Eva, welcome. So delighted you could join us today.
Thank you, Tiffany. Thank you for your kind introduction. And thank you also for inviting me to this DNA dialogue. I'm excited to present to all of you a uniquely Filipino disease.
And a special welcome to your colleagues from the Philippines who are joining us today as well.
Yes, thank you. They're all here giving me support. I hope you can see my slides now.
That looks excellent. I look forward to chatting to you after your presentation.
Thank you. So good afternoon. I would like to share with you our project on investigating the genetic epidemiology of X-linked dystonia Parkinsonism. This presentation covers the following: the discovery of X-linked dystonia Parkinsonism in the Philippines, the unique phenomenology and natural history of XDP, the genetic basis of XDP, what is known at the current time, investigating the genetic epidemiology of XDP, which is the focus of our project, the important role of genetic counselors in the care of XDP patients and families, and I'll just briefly talk about the Collaborative Center for XDP at the Massachusetts General Hospital, Harvard. These are the early drawings of Dr. George Viterbo from Rojas City, which illustrated a movement disorder first reported in literature only 40 years ago and was then called Dystonia musculorum deformans. In this landmark article, Dr. Lillian Lee, a Filipino neurologist, described 28 adult males who presented with torsion dystonia, and 23 of whom were from the island of Panay. There was no male-to-male transmission, so sex-linked transmission was inferred. Pedigree analysis revealed families with several members affected, and they were all males. Noteworthy was the observation also of Parkinsonian features in some of the patients, as well as their relatives. But where exactly is the Panay Island in the Philippines. So the Philippines is an archipelago consisting of 7,641 islands with three major island groups, Luzon, Visayas, and Mindanao. So currently I'm here in Manila, which is in Luzon. Panay is a triangular island located in the western part of the Visayas, and it's about 160 kilometers across. It's divided into 4 provinces named Aklan, Antique, Capiz, and Iloilo, all in the western Visayas region. Panay is the gateway to the most famous of white sand beaches and of the Philippines. And Boracay is the most famous one, which is at the northern tip of the Panay Island. It is famous for its pristine powdery white sand and clear turquoise water, and has been consistently ranked as one of the best beaches in the world. So I hope you can visit this place sometime. But what is X-linked dystonia Parkinsonism? It is a severe neurodegenerative disease that has arisen uniquely within the Philippines. It affects individuals with maternal ancestry from Panay Island, and the general population prevalence that has been computed previously is estimated at 0.34 in 100,000. However, the prevalence rate in the Panay Islands is 5.74 in 100,000, with the province of Capiz having the highest rate. Patients with the disease exhibit symptoms of focal dystonia in their 3rd to 4th decade of life, progressing to generalized dystonia and the appearance of Parkinsonism. A registry that was completed in 2010 identified 505 cases with 500 males and 5 females. So this was the first of the XDP Philippine registries. Of course, after that, we have a more recent registry, as many of the patients have already passed on. What the registry has gathered is that majority, about 94% of XDP cases present initially with focal dystonia, and only about 6% show initial Parkinsonian traits. Focal dystonia presents most frequently in the lower extremities in 31% of the patients, followed by the craniofacial area in 28%, the neck and shoulder area in 23%, upper extremities 14%, and the trunk as a initial presentation of the focal dystonia occurs in 2.6%. For lower extremities, focal dystonia often presents with dorsiflexion of the big toe, foot flexion, foot inversion, fanning of the toes, tremors of the feet and legs, leg spasms, gait dystonia, or lower leg dystonia. Some other descriptions in the other areas of the body. For the craniofacial area, dystonia will manifest with blepharospasms, twitching of the face, spasm of the face, jaw opening, jaw closing, mouth pursing, snout-like movements of the lips, tongue protrusion or retraction or rolling, and oromandibular dystonia. For the neck area, they can present as anterocollis, retrocollis, torticollis, and mixed forms are common. Sometimes even just neck stiffness or even tremors in the area of the neck. The upper extremities can present with writer's cramp, dystonic fingers and hands, and myoclonic and coriform movements have also been observed. In the trunk area, abdominal contractions may be the presenting symptom. There can be flexion at the pelvis, and torsion and extension of the trunk are the main manifestations. So in contrast to the typical adult onset dystonia, where the dystonia remains focal or at the most becomes segmental, in XTP, the initial focal dystonia spreads in 97% of the time, and generalizes within 5 years in 84%, regardless of the initial site of involvement. Those who start with lower leg dystonia appear to generalize earlier. So as the disease progresses, the predominant picture is one of nearly constant severe dystonic movements, usually affecting the axial areas with prominent oromandibular involvement such as jaw opening and tongue protrusion, as you can see in this drawing. At the height of the severity of the dystonia, it is difficult for the patient to assume any comfortable position. But nonetheless, despite the contortions, the patients often manage to move about, though in a quite bizarre fashion. The Parkinson's phase typically manifests with resting tremor, micrographia, bradykinesia, hypomimia, and shuffling gait. Maybe to better illustrate what I have mentioned in the last few slides, I'm going to share with you this video. So here you can see the patient having tongue protrusion. And his wife is taking care of him. He cannot, this is the bizarre dystonic movement, and he has to be fed because he cannot do activities of daily living anymore. So it says here, because his condition was getting worse, the Botox injections were not working. This picture shows him about just a year before the symptoms manifest.
Very good.
So this slide shows us the different phases of X-linked dystonia Parkinsonism, where you see the dystonic phase, the jaw opening, and the trunk dystonia. On the left, on the right side is the Parkinsonian phase, the right picture, I mean, and in the middle is the combined dystonia and Parkinson's phase. What we know so far is that any male, anywhere in the world, who presents with a combination of dystonia and Parkinsonism, for sure is Filipino and has maternal origins in the Panay group of islands. This is the first genetically supported case of X-linked dystonia Parkinsonism, or LUBAG. It's also known as LUBAG locally, reported in an Australian hospital. And I found this as a case report, but you can see here that this 47-year-old male who's from the Philippines comes from Negros, and his most likely his mom is the maternal roots of his mom. Now, what about XDP in women? This slide shows the picture of a 57-year-old woman who developed leg dystonia at age 50, followed by generalization of dystonia, including speech involvement and signs of Parkinsonism, generalized rigidity, bradykinesia of the hand and finger movements 2 years later. Her mental status remained intact. Her family pedigree showed she has several affected brothers, and extensive gene dosage analysis revealed that the patient actually has X chromosome monosomy in a subset of her cells. So her karyotype was a 45X, 46XX. And upon phenotypic reevaluation, the patient showed several clinical features of Turner syndrome, suggesting atypical Turner syndrome. And you can see her height compared to her brothers. And this case contributed to the first molecular explanation for the manifestation of an XDP phenotype in women. What is known currently about the genetics of XDP is that it's probably a founder mutation, and early studies revealed that individuals shared a set of 7 variants annotated as disease-specific sequences, so disease-specific changes, I mean. So DSC1, DSC2, DSC3, DSC10, and DSC12. The haplotype also contains a 48 base pair deletion and an SVA type retrotransposon insertion within an intron of the TAF1 gene. The puzzling aspect of this haplotype was that it appeared to be consistently, remain intact during inheritance. And there were no observed recombination events producing partial haplotypes in XDP individuals. The variants were also never detected in unaffected control subjects. So the 7 sequence variants all cluster in and around the human TAF1 gene, which encodes the TATA binding protein associated factor 1. And this is a core subunit of the TF2D complex, which is part of the general transcriptional machinery. In patient cells, this SBA disrupts normal mRNA splicing to decrease levels of the full-length TAF1 transcript. Now, further studies done by Dr. Bragg from Massachusetts General Hospital, and they reported this in 2017, they detected polymorphic variations in the SVA retrotransposon. They found the hexanucleotide repeat for those affected with XDP. The number of hexameric repeats among affected individuals increased the increase was strongly correlated with earlier age at onset. And you can see that here, where we can see that the length of the repeat correlates inversely with the age of onset. This slide illustrates the genomic segment previously associated with XDP on the long arm of chromosome X and HG19 coordinates. The 7 known XDP-specific variants comprise the disease haplotype are present here in the red box region. And the haplotype variants are there, the five disease-specific changes, 1, 2, 3, 10, and 12. If the 48 base pair deletion here, and here is the presence of the SVA type retrotransposon insertion. There are 8 genes shown within the broader linkage region, which includes the TAF1 gene. The canonical exons of TAF1 and the relative position of the SVA retrotransposon is shown here, and the domain structure of the SVA consisting of here the hexameric nucleotide repeats of variable length, the CCT-CT. This test that we are able to do at the Institute of Human Genetics determines the presence of the SVA retrotransposon insertion in intron 32 of the TAF1 gene. A PCR product of 3229 base pairs is amplified if the insertion is present. Subsequent restriction fragment length polymorphism analysis using a restriction enzyme gives 2 products, and this actually confirms the existence of the insertion. So how does XDP affect patients and their families? This typically presents in adulthood, and the disease severely limits the productive years of those affected. The average age of onset is about 39.7 years. It's a progressive disease with patients becoming more disabled, immobile, and dependent on others for care, particularly towards the late stages of the disease. Unfortunately, treatment is very limited to symptomatic management, like giving some medicine for the dystonia or for the Parkinsonism. But studies have reported promising effects of bilateral deep brain stimulation in mitigating hyperkinetic features of the disease. There is no known cure at the moment. XDP has a huge burden, not only to patients, but also to their families because of the loss of quality of life and productivity, the stigma, and genetic transmission. So we embarked on a project to address the gap in knowledge, and this is primarily the actual disease frequency being unknown. So we did a project to provide a comprehensive picture of the prevalence of the genetic cause of XDP in the country. And this is to provide crucial data for the development of health services and allocation of resources that can improve the quality of life of those families affected. Through collaboration with the Collaborative Center for XDP at MassGen, we were able to develop a rapid genotyping assay, which uses residual dried blood spots. And we were able to do this in a semi-high throughput genotyping manner to determine the national prevalence of XDP. With the use of anonymized residual dried blood spots from our National Newborn Screening Program, we were able to get nearly complete, unbiased representation of the Filipino population. We randomly selected 110,000 de-identified male newborn residual dried blood spots. We performed genotyping, followed by haplotype confirmation, and an estimation of the national prevalence of disease could be made. So to do the genotyping, this is just to describe the method briefly. We looked into additional sequence variants that flank the repeat expansion in the TAF1 gene region that are always co-inherited together. These markers were evaluated as candidates for a rapid genotyping assay, which is based on a readout on quantitative PCR. So one of the markers tested is a single nucleotide polymorphism that can be used and detected by qPCR. One primer pair detects the normal wild type allele and is coupled to a fluorescent dye, whereas the other pair actually detects the XDP allele and is coupled with another fluorescent dye. The assay detects the ratio of the wild type and XDP alleles to distinguish the hemizygous XDP males with one chromosome bearing the XDP marker, the heterozygous female XDP carriers, and one which just contains the wild type. So we were able to, this figure on this slide shows an example of the assay run on the DNA obtained from individuals in each of these three groups. So this was our general flow, and with this method, we were able to process about 13,000 samples a week to complete our study. And this is the result. From the randomly selected samples, we found that the prevalence of XDP is 61 per 100,000 males, which is much higher than what was previously reported. And as expected, it was highest in Western Visayas, where the Panay Island is, followed by 4B. 4B is actually south of Manila. But although these are de-identified blood spots, we have no information about the maternal, where the mothers come from, but we presume they are from the Panay group of islands. The third is actually way down in Mindanao. And this can be explained by the fact that in the 1940s, the president of the Philippines promoted an increase in the population in sparsely populated areas of the Philippines, and there was a migration of people from Panay to Mindanao. And so one of the first settlements was in the Coronadal area. So we think that that's the explanation for that. And then, of course, also in Metro Manila, because of the migration that happens from people from Panay moving to Manila for work or for family reasons. Now, knowing this disease prevalence has opened a very important opportunity for our patients, and that is the development of health services and allocation of resources, such as for our PhilHealth XDP Care Benefit Package. In fact, just yesterday, I met with people from PhilHealth because of all the computations that we need to submit to them in order to provide the funds necessary for services, laboratory, and the care, overall care of patients with XDP, including putting PEG when they're not able to eat anymore, or providing them with Botox injections, as well as packages for deep brain, the DBS, to help in the dystonic movements. This cost-effective and scalable genotyping platform can be used with, we thought that this is XDP, just using XDP as a model. This type of genotyping platform can be used to advance our understanding of diseases that greatly impact the Filipino population, especially because we hardly have any prevalence figures for diseases in our population. I cannot overemphasize the important role of genetic counselors in the care of our patients with XDP. Our genetic counselors are doing so much more because the disease has so many issues for families and also the whole community. First is about the stigma of XDP. In the past, patients with XDP and their families had suffered from stigma because of the disfiguring movements they often manifest. Many patients were kept hidden, and as they caused embarrassment to their families, not having a good understanding of that it was a disease and not something mythical that's happening. Like the transformation into some form of what we call aswang, or probably you're more familiar with like a werewolf type of transformation. The knowledge, just similar to that. The knowledge that the disorder is inherited made it even worse, and some even changed their names. Others migrated away from the island of Panay, and many denied the existence of a family member affected by XDP. Even when they were no longer able to deny the occurrence of the disorder, they would admit only to having Parkinson's disease, which was a more socially acceptable condition. Now, this is a heartbreaking story of a family from Panay. This 4-generation pedigree shows 5 siblings of the this proband who committed suicide. A study in 2015 by Dr. Hamora and Dr. Lee, both movement disorder specialists, showed that the suicide rate for patients with XDP was about 10.8%, which is much higher than the suicide rates in the Philippines in the general population, which was then about 4 per 100,000. Death, as reported, were by hanging, self-starvation, organophosphate poisoning, and self-mutilation, death by hanging being the most common. Now, the proband faced many difficult life circumstances aside from death of his siblings. There were lingering concerns about risk of other family members, taking care of his 83-year-old ill mother, supporting his brother affected with XDP. They have limited economic means, and though he is a teacher by profession, he now has difficulty speaking due to uncontrollable jaw opening. So he actually expressed that he wished to have cancer rather than XDP, but subsequently he was diagnosed with nasopharyngeal cancer. So how heartbreaking could I mean, I couldn't think of any other word to describe what has happened to this proband. So our genetic counselors actually are very much in touch with these patients because they live within the community. I used to go to the islands by myself together with neurologists and movement disorder specialists to provide genetic counseling. We were seeing like 90 to 100 patients in one clinic day on a Sunday because we fly there and then just come back to Manila early Monday morning. Fortunately, two of our graduates of the Master of Science in Genetic Counseling, Nicey and Ben. They are both Hiligaynon speaking. That's the language in that area. Were able to help more families, patients and families by being there. And they could do this because they were the care is more organized now and intentional as they work as genetic counselors at the Sunshine Care Foundation. Because they're both nurses too, they provide a holistic care to the patients. And this is just one of their educational awareness materials that they have developed, and they use it for education and counseling. So over the course of time, emerging roles have been identified and eventually integrated in the provision of holistic care for patients and their families with XDP. The emerging aspects of genetic counseling were identified through a strong collaborative relationship with different health professionals as presented in this existing model of care, where they collaborate with neurologists and movement disorder specialists, those involved in the screening for symptoms and diagnosis. They also collaborate with, they also do community awareness campaigns to debunk stigma and myths about XDP, and there's active case finding. They facilitate and lead research efforts, implementing research groundworks from coming both locally and internationally. And they navigate patients towards other health services to provide holistic care and improve quality of life, and create healthy communities through family-to-family support, coherence of the community advocates through establishing proper channels of communication, and strengthening existing support groups. So this is now integrated, as I mentioned, in the community through the Sunshine Care Foundation Neurological Care and Research. And it's rooted in a holistic approach. And for the research part, this is in close tie-up with the Collaborative Center for XDP, which is an international consortium of scientists, physicians, and advocates working to advance research and clinical care for patients with XDP. The center is dedicated to support leading edge or cutting edge research into the mechanisms underlying XDP and expanding clinical care. And in order to build a broad coalition of researchers with diverse expertise across international borders to find a cure, there are 3 grant mechanisms offered: an investigator award, 2 years, exploratory pilot grants of 1 year of up to $100,000 US, and postdoctoral fellowships for a period of 2 years. So in summary, what I have presented is the discovery of X-linked dystonia Parkinsonism in the Philippines, its unique phenomenology and natural history, the what is known as the genetic basis of XDP at the moment, how we did our project on investigating the genetic epidemiology of XDP, and the important role of genetic counselors in the care of XDP patients and families, and just a brief description about the Collaborative Center for XDP at the Massachusetts General Hospital. So this is the work of an amazing team of clinicians, neurologists, movement disorder specialists, scientists, geneticists, genetic counselors, nurses, and community advocates. These are my collaborators for the genetic epidemiology study on XDP, and I'm grateful to all of them. So thank you very much for your kind attention.
Eva, thank you so much for an absolutely fascinating journey through your exploration with XDP. It's, I think, too, the perspective you bring that really emphasises the impact on individuals and families, and then the sort of community implications of this condition, was really brought to the fore. And now Panay's beautiful beaches are on everybody's bucket list. Thank you very much.
Yep.
So we've got a question from Barbara that echoes one that I was going to ask as well. Given that, the molecular mechanism you've described well, and the fact that repeat expansion is inversely correlated with age of onset and severity of presentation. Is this, I saw that on your genetic counselling roles slide, you referred to screening and diagnostic analysis. Barbara asks, can you comment on pre-symptomatic testing for males in a family known to have the disease, as well as carrier testing on if that's applied, and what are the implications for downstream service delivery if screening is introduced?
Well, thank you for that very important question. At the moment, we only do testing on symptomatic patients. We have no screening, except for the fact that they may actually come to our attention because they may come to clinic because they have a brother who is affected, and they know that they have a 50% a priori risk of being affected themselves, except that the onset is not until about 39 years of age. So we do not do any pre-symptomatic testing, but if there were tests done on them, it might have been in the context of a research. For carrier testing, that's also the same. So, carrier screening. So during the counseling session, we already emphasized that females who are the daughters of affected males, they actually are carriers of the condition.
Mm.
Unfortunately, because many of you might be aware, or many may not be aware of, the fact that in the Philippines, termination of pregnancy is not an option. So it might be a very difficult situation to address when we do screening, like even for the babies, or even in utero. So.
And fertility services like IVF and pre-implantation genetic testing is not broadly available.
It's not broadly available, but some patients have been able to access those. There are at least 2 centers I am aware of that can do pre-genetic testing for monogenic diseases. That's quite recently. Many years before, they go to places like Hong Kong, Singapore, Taiwan to avail of those services.
Yes, yeah. So we've got a question from John. Lovely presentation, ever if not sobering. So he asked about the repeat expansion within the transposon and the stability of that, and whether the size of the repeat. Oh yes, you did mention that the size of the repeat expansion correlates with severity and progression. And you don't undertake that genotyping assay for the purposes of prognosis?
No, not at the moment.
Okay.
Not at the moment. As I said, in the genetic epidemiology study that we did after, so we used that as a screening method, and then we did the haplotype confirmation.
Okay, great.
And that's where we got the figure of 61 in 100,000.
Yeah, which was quite a shocking figure.
Yes, we were shocked with that figure too, because we always felt it was like we considered it a very rare disease. But with that genetic prevalence study, it might not be as rare. However, we have not stopped yet. So after we completed our 100, the total from the 110,000 was actually only 107,000. We are now moving into getting 150,000 more cards. to complete 250,000, it might give us a more accurate figure of the prevalence. And after that, then we will be publishing this report.
Gee, the lab's working hard with 13,000 cards worked through a week, was it?
It was, yeah, around that.
Extraordinary. So Mohammed asks us a question. Thanks you for the presentation. He's asking about the other 4 variants, aside from the SVA insertion. Are they merely part of a co-inherited haplotype region, or have they been shown to play a molecular role in XDP pathogenesis? So is there a functional study, or have there been further investigation as part of the international consortium, perhaps?
Not that I am aware of. Unfortunately, there are many ongoing studies now, and until such time that the projects are completed, then we will know better. I am not aware of the disease-specific changes having any functional value at this time, or at the moment, it's really the target is really the SVA retrotransposon, causing a disruption in the transcriptional machinery.
Okay.
But I actually made a presentation about the CCXDP, the Collaborative Center, because they make a yearly call.
Oh,
And
great. All right.
I'm not sure, I have attended many of those meetings, and we also make our presentations in the beginning for the clinical presentations like natural history. Now, of course, last year, we presented our initial studies on this rapid genotyping assay. I have many of the scientists I meet there are from Europe, from the US. I haven't met anyone yet from Australia. So, they're opening these calls for everyone.
Fabulous. So functional genomicists from Australia should pay attention to the call. Keep an eye out. Okay. So Maddie asked a question asking about the potential for gene therapies for XDP. Is that something that's on the horizon, and what's the implications or potential for these families?
Yes, at the moment, I am aware of discoveries for drug repurposing, but although it has not reached us yet. So in terms of like the clinical trials, that should be done when there are good candidates already, which has reached that part where it can already be put on clinical trials. But for gene therapy, there's some work on CRISPR in the TAF1 gene, but whether it has progressed, I don't think I remember very well whether it has reached that point where it can be a good way of reaching therapeutic value.
Yes.
Yeah, potential. Yeah, but CRISPR has been there is a publication on that.
Okay, great. Eva, the epidemiology study that you undertook clearly was very valuable in terms of advocating for reallocation and prioritization of health budget and service delivery. Given the impact and severity of these conditions and the impact on the family in terms of carer burden, you mentioned that teacher that's sort of multi-generational care, as well as inability, like the economic impost of this condition on the entire family. Have you had any success in advocating for social support, or funding to these families that have a heavy carer burden?
Yeah. So that's a very important question, because you have to, if we think about it, these are men in their most productive years, because it's between 35 and 40 when the symptoms manifest, right? So some of them, actually, many of them are seafarers. There are certain parts of the Philippines where a number of their male providers for the family are seafarers. So just to tell you one story, wherein one of the patients who started showing the dystonic manifestations while he was on board the ship. He was brought down in Malta, and he was treated for meningitis. So after the course of the antibiotics, he continued to have the dystonic features. So he was flown back to Manila. And of course, the referral to specialists were already made, and they looked at the family history. So in other words, he was diagnosed with XTP. So these people who are working, productive, providing for their families, are now left with no work and needing, care for their activities of daily living, just like the video I showed you about Cromwell, who just 1 year ago was very able, good body size, able to
Yeah, it was a
work.
shocking decline.
Yes. And now completely under the care of his wife. So wives are the main caregivers. Sometimes if they're single or their wives have left them for some reason, it's their mothers who take care of them or their daughters. So through the Sunshine Care Foundation, and even actually before that, there were already some small, in the beginning, small projects related to like some form of industry where these people can like work at home while taking care of their husbands or their children or their male children who are affected if it's a mom. And also when the Sunshine Care Foundation came to be in 2014, I think that was the time that they were already, we call them community advocates or CA. So they're the ones helping each other out. It has to be a whole community to help each other out.
And the destigmatization and education, I guess, is such a core component of that.
That's right. I used to go to the communities, as I mentioned. They will hide their affected members. They will not take them out. They feel that there's something that happened to them, which was more related to something mythical. They were cursed or something. But then now, because they go to the clinics and they follow up, they're given care. Although the foundation, of course, does not have limitless resources or unlimited resources.
Yeah.
So there has to be some form of sustainability for the foundation to carry on its tasks. I don't know if you remember, I forgot to mention in the picture that I showed earlier that the genetic counselors even have to ride, they walk through the rice fields, go up the mountains, or like go through the river system, riding those rafts made out of bamboos to reach the patients.
Amazing. Very dedicated people.
Yeah.
So we've got a question from Miller about the Centre for Population Genomics in Australia. So they're conducting a genomic research that's collecting blood samples from the Filipino community in Australia. And Miller is wondering whether your work in any way could benefit from inter-country sort of collaboration with this research. She notes that there's a large Capizeno Club in Melbourne and Ilongo Association in Sydney and Melbourne. So that could be leveraged perhaps.
Of course, yes. And you mentioned Ilongo. Ilongo is the group of people who come from the Panay group of islands.
All right, so that could be a very useful connection
Yes,
then. All right,
yes.
I'll put you in touch with Miller and Daniel.
Thank you.
You did mention too, Eva, that the high throughput genotyping platform that you've established for this epidemiological study that could potentially be leveraged by other, to examine other conditions of higher prevalence or significance or priority conditions, genetic conditions. Can you speak to any of those? What's your future directions in that space?
That's again a good question. Right now, I'm working with a lot of cardiologists, and as I said, we don't even have prevalence figures for like familial hypercholesterolemia,
Oh, really?
Yeah. So, I mean, we know the worldwide figure, but we don't know how common it is in the Filipino population. So there are so many, the best example where we were able to get prevalence figures for conditions is related to our newborn screening program for our metabolic conditions, like also for congenital hypothyroidism. But for many adult diseases, we don't have any. We always just say in the US, the prevalence is like this.
One
In
in
Japan,
240. Yeah.
yes. So we are in the planning stages for that because one important information is the genetic basis is known and that we can find useful candidate markers to run into a genotyping platform. That would be
Excellent.
important before we could actually, we have to choose the disease that we will be able to run in that platform.
Yep, understood. Understood. Well, Eva, we've got no other open questions. It has been such a pleasure speaking with you today, and it's lovely to see you again. And we certainly look forward to sort of hearing about your work moving forward in the future. And I look forward to catching up with you at some conference somewhere in the near future.
And thank you again for having me here to share with you this uniquely Filipino disease called X-linked dystonia Parkinsonism. And I think I really would like to extend that invitation for anyone in your group who would be interested to work on this, because you're actually looking at striking you'll strike two diseases at the same time. Dystonia and
And
Parkinson's,
Parkinsonism.
right?
For
So
sure.
I think we can all work on this together, and that's the aim actually of the Collaborative Center for XTP.
It's a wonderful call to action. So thank you so much, Eva.
Thank you. Bye-bye.
Bye.
Thank you.
And thank you all for joining us for DNA Dialogue today. I do hope you can make our final DNA Dialogue webinar on the morning of the 17th of June, when Heidi Rem will be presenting, as she did for our very first DNA Dialogue in May 2022. And like that webinar, Catherine North will also be joining us for on the 17th to farewell this series and Australian Genomics. So I hope you can join us then. Thank you. Bye-bye, Eva.
Bye.